Globally, over 450 million people have diabetes, and this number is projected to rise over the coming decade. Type 2 diabetes accounts for over 90% of diagnoses worldwide, and current management guidelines for type 2 diabetes prioritize the use of glucagon-like peptide-1 receptor agonists, or GLP-1RAs, in specific populations and before insulin therapy. Semaglutide is a medication that binds to and activates the GLP-1 receptor. It was shown to be both effective and safe in treating type 2 diabetes in 10 clinical trials known as the SUSTAIN trials. These trials included a heterogeneous population of patients with type 2 diabetes. Therefore, it would be helpful to know whether semaglutide is equally effective and safe across all subgroups of patients. A new post-hoc analysis re-examined the Phase 3A data from the SUSTAIN 1-5 trials to assess any differences in response to semaglutide among patients with different clinical baseline characteristics, such as HbA1c levels, background medications, diabetes duration, or pancreatic beta cell function. The researchers found that those with the poorest pretreatment glycemic control, reflected in the highest initial levels of HbA1c, saw the greatest improvement over the 30 weeks of semaglutide treatment. This resulted in similar post-treatment levels of HbA1c among all groups. In contrast, reductions in body weight were greater for those with lower initial levels of HbA1c. Compared with treatment with semaglutide 0.5 mg, treatment with semaglutide 1.0 mg resulted in greater improvement in both HbA1c levels and body weight, independent of any patient differences such as background medication, diabetes duration, or pancreatic beta cell function. Semaglutide was found to be safe across all patient subgroups, with similar, mostly mild adverse events occurring in all subpopulations of patients. These results show that semaglutide is both effective and safe for treatment of type 2 diabetes in a broad spectrum of patients with a range of clinical baseline characteristics.